Okay, you can start.
Topic for the today's session is Staphylococcus. First one is the introduction. Staphylococcus is a gram-positive cocci. It is classified into two families, MICROCOCCACEAE family and STREPTOCOCCACEAE family. First one is the MICROCOCCACEAE family. The example of MICROCOCCACEAE family is Staphylococcus and Micrococcus. It is catalyzed positive. Arrangement in gram stain. Gram-positive cocci is arranged like a cluster-like formation in Staphylococcus, and gram-positive cocci is arranged like tetrad-like formation in Micrococcus. Next one is the Streptococcaceae family. The examples are Streptococcus, Pneumococcus, and Enterococcus. It is catalyzed negative, and gram-positive cocci is arranged like chain-like formation in Streptococcus, and gram-positive cocci is arranged like pair in Pneumococci and Enterococci. First one is the Staphylococcus aureus. It is catalase positive, coagulase positive, facultative anaerobe, non-motile, non-sporing, and occasionally capsulated. In Greek, the Stephel means bunch of grapes. From this picture, we can see the appearance of the grapes. Next one is the virulence factors of Staphylococcus aureus. First one is the cell wall factors, peptidoglycan. The activity of peptidoglycan is it is more thicker and it confers cell rigidity and induce inflammatory response. Teichoic acid. The activity of teichoic acid is it helps in adhesion to mucosal surface and prevent opsonization. Next one is the clumping factor or bound coagulase. It is responsible for slight coagulase reaction. Next one is the protein A. The activity of protein A is antiphagocytic, which means it prevents the action of phagocyte, and anti-complementary. It prevents the with the antibody. Next one is the toxins. First one is the membrane active toxins, hemolysin, alpha hemolysin. It is inactivated at 70 degrees Celsius and reactivated paradoxically at 100 degrees Celsius. Leucocidal cytotoxic, dermonecrotic and lethal. Next one is the beta hemolysin. The activity of beta hemolysin is pingomyelinase. It lyses sheep RBC, but it will not lyse the human RBC or rat RBC, and it will exhibit the hot cold phenomenon. Gamma hemolysin. It is a bicombinant protein and it will lyses the rabbit, sheep, and human RBC. Next one is the delta hemolysin. It is detergent like it will lyses rabbit, sheep, and human RBC. Next one is the Phantom Valentine toxin. It has two components, F and S. It will damage the polymorphonuclear lymphocyte and macrophages. It is associated with community-acquired methicillin resistant Staphylococcus aureus syncohymenotropic toxins. It is a bicombinant toxin such as gamma and pentanvalentine toxin. It acts synergistically and are called synergohymenotropic toxins. Other toxins such as epidermolytic toxin. The other name for epidermolytic toxin is exfoliative toxin. It mainly belongs to phage group 2 and it will cause scalded skin syndrome in which the epidermal layer is separated, and severe Reiter disease. It will occur in newborn, and toxic epidermal necrolysis. It will occur in adult. Milder pemphigus neonatum bullous impetigo will occur. Next one is the enterotoxin. It is produced by 50% of clinical isolate and it will cause food poisoning. The incubatory period of enterotoxin is 1 to 6 hours due to performed toxin. Site of action. The toxin will stimulate the vagus nerve and vomiting center of the brain. It also stimulates the intestinal peristaltic activity. It is a heat stable, and most common food items involved are milk products, bakery food, custards, potato salad, or processed meat. It is a multiple antigenic type. Most common type is type A. Next one is the toxic shock syndrome toxin. Most strains belongs to phage group 1. Enterotoxin F. It is the most common toxic shock syndrome toxin, followed by enterotoxin B and C. Risk factor of toxic shock syndrome toxin is by using the vaginal tampon by menstruating females. Males won't get affected. Anti-toxic shock syndrome toxic 1 antibody is protective. Manifestations: rash, fever, hypotension, and multi organ failure will be there. Diagnosis of toxic shock syndrome toxin: detection of toxic shock syndrome toxin by latex agglutination test and enzyme immunoassay. Detection of toxic shock syndrome toxin gene 1 and 2 by PCR. Treatment of toxic shock syndrome toxin is clindamycin. It will reduce the toxin synthesis. Next one is the pathogenesis: skin and soft tissue infections. Bacteriomycosis, it is a mycetoma-like condition. Tropical pyomyositis, osteomyelitis, and septic arthritis. The most common site is knee. And post-operative parotitis, parenchyma, pyomyositis. Nematocele, it is shaggy and thin-walled cavities in lungs which will occur in neonates. Abscess, psoriasis abscess, epidural abscess, surgical wound infection, folliculitis, inflammation of follicles, pharyngeal, carbuncle, and hidradenitis suppurativa, mastitis, and breast abscess in nursing mothers. Toxin-mediated disease such as toxic shock syndrome, food poisoning, scalded skin syndrome will be there. Infection associated with community associated methicillin resistant Staphylococcus aureus. Skin and soft tissue are the most common site for community associated methicillin resistant Staphylococcus aureus. 5 to 10% of strains are invasive and it will cause various invasive infections such as necrotizing pneumonia, sepsis with water hose, Friedreichsen syndrome, or purpura fulminans. Streptococcus aureus is rare cause, most commonly caused by meningococci. Necrotizing fasciitis. Staphylococcus aureus is a rare cause. Streptococcus pyogenes is the most common cause. Next one is the lab diagnosis. Under direct smearoscopy, pus cells with gram-positive cocci in cluster. Under culture, nutrient agar golden yellow pigmented colonies will be seen. In blood agar, colonies with narrow zona beta hemolysis. Selective media are mannitol salt agar, salt milk agar, Rutgams media. Next one is the biochemical identification. It is catalyzed test positive, test differentiating staphylococci from micrococci. It will show a fermentative pattern. And test differentiating Staphylococcus aureus from coagulase negative Staphylococcus. Staphylococcus aureus is positive and coagulase negative Staphylococcus is negative. Coagulase test slide and tube is positive. Heat stable thermal nucleus test is positive. DNA test will be positive. Phosphatase test will be positive. Mannitol sugar is fermented. Black colored colonies on potassium tellurate agar. Gelatin liquefaction is positive. Protein A detection will be there. Next one is the typing of Staphylococcus aureus. Most common method of typing Staphylococcus aureus is phage typing, which means a pattern method. Epidemic strain of Staphylococcus aureus is phage type 80 by 81. It causes outbreaks in hospitals. Next one is the Staphylococcus aureus carriers. About 25 to 50% of healthy population are carriers of Staphylococcus aureus. Most common site of colonization is anterior nose, which means anterior part of the nose and skin, perineum, axilla, and groins. Most common way of spread of infection in hospital through the hands of hospital staff. Most effective way to prevent the hospital infection is by hand washing. Next one is the coagulase negative Staphylococcus. They are the mostly the normal flora of the skin. Staphylococcus epidermis, most common coagulase negative Staphylococcus, accounts for 60 to 70% of coagulase negative Staphylococcus. It will produce polysaccharide, glycocalyx, slime, biofilm production. It will adhere to the implant of foreign bodies like valvular shunts and prosthetic devices. Infections such as endocarditis with the insertion of valvular prosthesis, ventricular shunt infection, and stitch abscess will be there. Next one is the Staphylococcus saprophytis. It causes urinary tract infection in young sexually active females. It is resistant to novavirus. Thank you.